This podcast service from News Talk WHCU is brought to you by Black Box Computer Consulting.
It appears as though the Force is now with us from Cornell University. The future of prostate cancer therapy may lie in a tiny, sticky silicon chip. It's been dubbed the Jedi. It's an acronym. We'll explain. I actually am not bright enough to explain, but I know somebody who is, our next guest. He is the Associate Professor of Mechanical and Aerospace Engineering at Cornell University. Brian Kirby joins us this morning. And for starters, Brian, what is an Associate Professor of Mechanical and Aerospace Engineering doing with looking at prostate cancer?
Well, good morning. My expertise is really fluid mechanics, and so that's why I work in a mechanical engineering department. But it turns out that if you're trying to learn about cancer and you want to do it by looking at the peripheral blood or the blood that you take out of a patient's arm, what you really need to do is find some way to sort out and find the cancer cells, which are very rare in blood. So, and that ends up being a fluid mechanics problem.
A food mechanics problem. I've never thought of my food as mechanical before.
A fluid mechanics problem.
All right. So, Brian, what does JEDI stand for?
So JEDI is an acronym that stands for Geometrically Enhanced Differential Immunocapture. And what it really means is that we're using antibodies to capture these cells that are very specialized. But we don't just use the antibodies. We also use a microfluidic device that has a geometry that's designed to cause cancer cells to collide with the surface very often and to prevent the other cells in the blood from colliding with that surface. And by combining both a surface that's sticky to a specific sort of cell with a geometry that makes sure that that cell collides with the surface, we're able to capture these relatively rare cells with a high purity.
So you are capturing cancer cells with like this nanobot that you put into the bloodstream?
It's not something we put into the bloodstream, but it means that we can take blood directly out of someone's arm, and then we can run that small amount of blood through a microfluidic device and identify these cells. And the reason why these cells are hard to get is that in 1 milliliter of blood, you have about 5 billion cells. And a late-stage metastatic cancer patient may have 1 or 5 or 10 or 20 cancer cells inside that. So we need to find this one cell out of 100 million or a billion cells.
And this new JEDI chip has gone far beyond that, as I understand. Now, metastatic cancer, I get. It's the cancer that spread from the place where it first started. And in the case of what a team of researchers at Weill Cornell Medical College in New York City and Cornell's College of Engineering in Ithaca have built this chip into a device that captures an unprecedentedly high concentration of rare cancer cells from this metastatic prostate cancer patients. And what is the purpose and potential of doing such a thing, Brian?
Well, the key, one of the challenges associated with cancer is that there's a lot of information you can get from the cancer tissue. And so if you remove the primary tumor from a patient, you can learn a lot about that cancer. But oftentimes, as the cancer evolves, the cancer has really changed from that first version of the cancer that happened when a primary tumor was removed. So if a late-stage prostate cancer patient is undergoing metastasis, the nature of those cells that are forming those secondary tumors is often different from those of the primary tumor that may have been removed a year ago or 5 years ago or 10 years ago. So we need to get information about those secondary tumors. And the way we do that is by capturing these rare cells. Now, the potential is that if we can create this sort of window into the cancer process or this metastatic process, we can allow clinicians to immediately make sound decisions about how to treat the patient. So, for example, we can help them identify which chemotherapeutics are most likely to help the patient.
So you can begin to individualize treatment based on what you know about these metastatic cancer cells that now this JEDI chip, GEDI, is able to take blood, pump through a nanoscale channel filled with little posts that are sticky and are able to attract these, what you called, Brian, needles in a haystack.
That's right. And the key thing, if you think of a specific example for prostate cancer, we've been able to capture these cells, and then we've been able to do functional analysis of them. And what I mean by that is that we've been able to expose these cells outside the body to the chemotherapeutics that might be used on the patient. And in a paper we recently published in PLOS One, we were able to show that by measuring the response of these cells, that this then gave us information about how the patients would proceed if they in fact took those drugs.
And the paper that you published in PLOS ONE has been peer-reviewed now. This has become the breakthrough science in cancer research, as I understand it. But all science, Brian, starts with a hypothesis. And so this is obviously built on generations of cancer research and generations of support, as this comes in part through a National Institute of Health-funded Physical Sciences Oncology Center grant. So this is research that is, I would say, you'd figure somewhere in the middle of the road as you are going down this long road towards treating, if not finding a potential cure for cancer. Where do you find yourself in the whole grand scheme of cancer research right now? Obviously at the forefront, but how much further do we have to go?
Well, I think cancer is a whole family of diseases with all sorts of different problems, ranging from how we detect cancer early to how we properly treat cancer, how we stage cancer, et cetera. In late-stage cancer, which is what this work has been primarily about, it really gives us the potential to intelligently manage cancer. So if a patient has had cancer for some time, allowing us to choose the right drugs to treat them can be very useful. It has other applications, and those other applications are also very exciting as well. And the same sort of approach that we're using now, we think has potential to detect cancer early in some specific cases.
Wow. And
so that's something that we're working on quite actively. And, as you said, this work is all partially supported by this Physical Sciences and Oncology Center, which is a large collaborative effort combining people at Cornell Ithaca and also Weill Cornell Medical College.
Associate Professor of Mechanical and Aerospace Engineering, Brian Kirby, joining us this morning in the very latest. You just... Sir, may the force be with you.
Thank you so much. Have a good day.
Thank you, Brian.